My great pleasure today to introduce Yatendra Kumar to our session. So Yatendra started his journey in science, studying life science and biotechnology as bachelor and master degrees, and then he did a PhD in global regulation on gene transcription in yeast in Hyderabad, where then he also did a short period as senior project fellow, analyzing the role of histone modification in adult neurogenesis. After that, he moved to Europe, to University of Edinburgh, in the lab of Wendy Bickmore, where he's studying the role of histone globular domain acetylation in gene regulation. Yatendra, we're very happy to have you here. I'm really looking forward to your talk. The floor is yours.
Thank you. Thank you, organizers, for having me here and for the kind introduction. And I'm really glad that I'm going to give you a story here, which actually involves fragile nucleosomes. It revolves around that concept. So I'm going to talk to you about the acetylation of the lysine 115 in histone S3, which happens to locate itself on the nucleosome dyad. And to start, in general, what we see is that histone acetylation is associated with gene activity, and subsequently, many of the N-terminal many of the lysines in the N-terminal and their acetylation state has been used as a marker of active chromatin. For example, H3K115 acetyl mark, which is a very popular mark of active enhancers in mammalian genomes. So the tail domain modifications can create these docking sites for other effector proteins to bind and affect their remodeling activity, for example, on the chromatin. Apart from this, the tail domain modifications can affect internucleosomal interactions, and thus they contribute to the transcriptional state or the regulation that goes on via epigenetic mechanisms. Apart from this, we have globular domain in the histone, and there are lysines in the globular domains. And they are important in the sense that they can have, they can affect the modification. For example, acetylation can affect the DNA histone interactions and thus help regulate the wrapping and unwrapping of the DNA from the nucleosome particle. Another part is that these modifications can affect the histone, the code itself, the nucleosome core itself, because they can then have some destabilizing effects because of the static hindrance. So we are interested in exploring the role of globular domain modification, especially lysine acetylation. And in this direction, previously we studied H3K122 acetylation mark, and we found that this mark can be used as a mark of active enhancers independent of H3K27 acetyl mark. So there's a set of enhancers which are missed out if you simply rely on K27 acetyl mark. This does highlight the importance of globular domain acetylation, and there is so much to be learned on these modifications in the globular domain of histones. So in the same direction, we are focusing on H3K115. Now this lysine is very interesting because it is exactly located at the surface where you have dyad axis passing. So you can see in the nucleosome structure the dyad axis, and if you turn in the middle, that when you are looking down the dyad axis, you can see that both lysine residues are from two H3 molecules. They come together, they are juxtaposed very close within the region where you have the central base pair of the nucleosomal DNA. Now the dyad axis is very important because it has very important DNA histone interactions, which help stabilize the DNA on the histone octamer. So in that sense, it can affect nucleosome dynamics even more because these two residues, they come together and the effect of acetylation would be double. Now, this is supported by the in vitro experiments in the field, where nucleosome assembly in vitro was used to show that the octamers containing H3K115 acetyl mark, they have lower affinity to the nucleosomal DNA compared to other marks such as K122 acetylation. This means that acetylation at this location affects the stability of DNA histone interactions because acetylation takes away the positive charge from the lysines. So with this supportive evidence, we wanted to see how to explore how this modification plays out in vivo. So with that in mind, we performed native amylase ChIP-seq for H3K115 acetylation mark in mouse ES cells. And to the left, what you're seeing is the correlation matrix of ATAC-seq along with ATAC-seq and K115 acetylation along with other tail domain modifications. As you can see, K115 acetyl regions, they cluster together with ATAC-seq compared to any other tail domain modification, including K27 acetyl mark. In this sense, this K115 acetyl, as expected, shows strongest correlation with open chromatin. And then we used Rome HMM segmentation of H3K115 regions. And we found that this PTM, a greater proportion of K115 acetyl peaks, is associated with active enhancers and promoters compared to two other histone modifications, K122 and K27 acetyl marks. So this has got a closer relationship to active chromatin, along with the more tighter association with chromatin accessibility altogether. Then we wanted to see how this histone modification changes during differentiation, for which we used mouse ES cells, and we differentiated them into neuroprogenitor cells in a 7-day protocol. And when we define promoters between day 0 and day 7, whether they lose K115 or they gain K115 acetyl mark. So promoters that lose are down-regulated gradually, while promoters that gain H3K115 acetylation at the end of the differentiation, you can see at day 3, 5, and 7, they are gradually upregulated. So K115 acetyl mark follows the, it kind of mirrors the transcriptional dynamics during differentiation also. So it seems like a functional mark from this perspective. And then we wanted to see how it looks on the enhancers. So as I told you before, we found that K122 acetyl mark in the past, it was present on a certain set of enhancers. And our enthusiasm was a bit dampened when we saw that there are no enhancers which are specifically marked by H3K115 acetyl mark. The enhancers that have these are the very strong enhancers, but they also have other acetyl marks. So it seems that while this mark is associated with high chromatin accessibility, it does not mark any specific set of enhancers. Things got intriguing when we looked at promoters. So in general, you see histone acetylation marks, they correlate with gene expression, but we also see that the K115 acetyl the average signal does not show this familiar dip which active promoters exhibit. So unlike K122 escalation and K27 escalation mark, K115 does not show the peak. So that's intriguing because we all know that the nucleosome occupancy at the active promoters is really low, and this is the same region where K115 escalation is peaking. It's not in the gene bodies, it's not in the upstream, it's on the region which is called nucleosome free region, which actually marks the remodeling of the promoted region. So this is intriguing because in this region there are no nucleosome, but you have a signal for a histone mark. So this brought us to the concept of fragile nucleosomes. But before that, let me summarize how is it that we have this common notion of a nucleosome-free region or nucleosome-depleted region on the active promoters. Most of the nucleosome maps which are generated in the field, they are coming from over-digested chromatin in the sense that most studies have used very high MNase digestion level and to a level where the majority of the DNA of the population is mononucleosomal, and this band is then taken out of the gel and then it's sequenced. So what's happening is that we are generating over-digested chromatin, and that is causing that is showing this profile. But if we use partial MNase digestion, or if we use lower salt in the chromatin fraps, we can see that there is a population of nucleosomes which are present in the NDR regions of the promoters, and they also correlate with gene activity. They are highest in the highly expressed genes. So these nucleosomes which are MNase sensitive and salt sensitive are called fragile nucleosomes altogether, and they are associated with the chromatin remodeling which is happening at active promoters and enhancers. Similarly, a subset of fragile nucleosomes can be present as subnucleosomes as they don't have a complete histone octamer, hence, and they show a smaller MNase footprint. So with that in mind, we redid our ChIP-seq, but in paired end mode, which allowed us to fractionate the chip fragments into sub and mononucleosomal ranges. And then we compared the AT content of these DNA sequences, which are pulled with K115 or K27. So both subnucleosomal as well as mononucleosomal signals, we compare that S3K27 and S3K115 escalation. The AT content is higher for the chip libraries in S3K115 than K27. Now, higher AT content is related to related to higher nucleosome turnover, which relates to indirectly to nucleosome mobility. Apart from this, K115 acetyl mark also picks up a smaller MNase footprint compared to the input libraries and compared to K27 libraries. So these are all the features of fragile nucleosomes. With that, when we look at the then we plotted S3K115 and K27 chip libraries along with input, and this time we are plotting different fragment lengths with respect to the distance from TSS. So the area between the two dashed lines is basically 0 TSS to -100 position in the upstream, and you can see that the K115 acetyl mark is present in this so-called nucleosome-free region, and S3K27 acetyl mark is depleted from this region, although it is present in the gene bodies. In the mean occupancy plots also, you can see that both sub and mononucleosomal signals are coming from K115. They are enriched just upstream of the TSS, while that is not true for S3K27 acetyl mark. Now we come to the enhancers. Why S3K115 acetyl mark? When we look at the enhancers on the left, these enhancers have two OCT4 motifs, just to signify the chromatin remodeling which is going on. At the center of the enhancers, you can see the ATAC-seq signal in the middle and canonical MNase nucleosomes in the sides. and H3K27 is depleted from this region of remodeling, while K115 acetyl mark is enriched there. Similarly, OCT4 and nano motifs, when they are present in the same enhancer or they are present alone, the enhancers with OCT4 alone have much lower signal for K115 acetylation, and compared to the double occupied or double acted upon enhancers. And K27 acetyl always shows this dip. So enhancer NDRs are depleted of K27 acetyl mark, but they are enriched with K115 acetyl mark. Now, another important class of regulatory regions is CTCF binding sites, which form the basis of genome folding in vivo. So we found that the subnucleosomal H3K115 signal is enriched exactly on the motif of the CTCF, while we have mononucleosomal K115 enriched in both sides of the CTCF motif. And in case of K27 acetyl mark, we have this marked depletion, which is in line with the nucleosome depletion NDR model of these sites. So I would like to summarize that as the K115 acetyl mark is a unique marker of chromatin accessibility associated with regulatory sites, and it mainly relates to the fragile nucleosomes, which are chromatin remodeling intermediates at these sites. In comparison, the other modifications, such as K27 acetyl mark, is present in the upstream and downstream nucleosomes, but not in the dead center of the remodeling. Then this is true for transcription factors and CTCF sites, as well as promoters and enhancers. All this indicates that this mark could be creating those fragile nucleosomes to facilitate chromatin accessibility, factor binding, transcription, etc. So it might contribute big time in chromatin accessibility, which is a must for activating chromatin. So what we are doing right now is that we want to have a direct evidence of this modification on subnucleosomes, which we are trying to do by sucrose gradient segmentation, and which can be used to isolate different nucleosome species. And then we want to probe for this modification and other modifications such as K27, and also for factor binding. And then we would do ChIP-seq on those different fractions to illuminate different species and its relationship with K115 acetyl mark in the genome. And we are also creating a conditional knockout line for H3K115 acetylation, for which we will use prime editing to convert all lysine 115 to arginines, which cannot be acetylated. And we'll keep rescue copy of S3.3, which would allow us to to avoid adaptation during the prime editing of this line. And then we can ask whether K115 acetylation mark is required for ongoing transcription, or transcriptional induction, or transcription factor binding. What is the role in CTCF binding and insulation, and how these nucleosomes carrying this mark interact with ATP-dependent chromatin remodeling complexes. With that, I would like to thank everyone in my lab, especially my former colleague Dipto, Elias, Luciana, and Rob Illingworth for their direct contributions, and our collaborators, Christian Helen, Chloe Stanton, and Nick Gilbert, and our funding bodies. With that, I would like to thank all of you for your kind attention and your feedback. Thank you so much.
Thank you very much. Very, very nice talk. Very clear and linear. I think Ben had a question.
Yeah, fantastic talk. I was wondering if you've thought about whether this K115 acetylation is able to is interacting and modulating activity of things like remodelers, and that's contributing to the subnucleosomes that you're seeing.
We haven't tested it yet because it's a bit difficult. First of all, we are focusing whether this is required at all or not. It has a very unique features, such as association with the fragile nucleosomes. But then if I can take you to the example of H3K27 acetyl mark, this was edited in mouse CSCs, and it was totally not required for ongoing transcription or enhancer activation, etc. So that is something that is, this redundancy is there in terms of, in case of histone acetylation. And they are not easy to modify just by inhibiting the enzyme, because then if you go after the head complexes, they are quite, they are not so specific. So we know that the P300 head deposits this mark, but P300 deposits so many other histone acetylation marks. So it's not, so we are not there yet. But once we have the line, we would like to know how remodeling is affected in the cell. Does that answer your question?
Yeah, absolutely. And like, I completely agree that doing the point mutant as the first experiment makes a lot of sense. Thank you.
Yeah, it's not an easy one because there are just so many copies with the histones.
Yeah. So we have then a question in the Q&A box. Actually, two. One is, sorry if you said it, but what is the enzyme responsible for K115 acetylation? And the second one is probably connected to what you just said. So how do you explain that the fragile nucleosome escapes the acetylation of K27? And could it be a matter of potentially lower residence time of the fragile nucleosome, and whether you look at that?
Perfect. So for the first one, we know that P300 is one of the enzymes that could do this, but we are testing other heads also. We know that P300 does it. When we inhibit P300, it reduces the levels. And second one, the nucleosome thing, that why K27 is not there. Right. My enthusiastic guess is that when you have these, transcription factors targeting the promoter or enhancer, the primary purpose of the heads which are recruited is K115 acetylation. But then the head activity diffuses here and there.
Okay.
Yeah, so this is my guess, but I still don't have the right answer for why those histones where we have K115 acetyl, there is no K27, which is also a point that I drive for the validity, because if I had K27 also everywhere here and there, then you would know that okay, the antibody didn't work or something. But here it's not the case. So totally, in the light of the redundancies that head complexes have, and there's so less known about it, we don't know how we have this mark, but not K27.
Okay. Mildly connected to that a bit more on the technical side. So you said that you can see the 115 acetylation with mild digestion with unfixed chromatin, basically. I was wondering whether you could see a difference, or maybe at this point even potentially a little bit of a rise of K27 if you tried to do that with fixed chromatin, because at that point you don't have the risk of losing the fragile nucleosome anymore.
We don't prefer this cross-link chromatin for histone modification. Somehow we get cleaner signals.
Yeah,
even for K27 acetylation, we have much cleaner libraries from native chip than this. So if it was there, because we are referring to a much rarer species of nucleosomes, so if it was there and we can see K115 acetyl mark, we should have seen K27 also, because that is much more, technically speaking, this is more abundant in the cell.
Yeah. So yeah, because I was thinking of something like, if you have a fragile nucleosome, maybe if you have 115 first, and then you add another acetylation, that at the point the residence time of that double modified nucleosome is very short, and at the point you lose it,
Yeah.
it's cheap, and that's why you don't see it. While potentially, although the signal, I agree, is much less clear, you could potentially see it in the
Yeah, totally possible. In terms of the
example.
residence time, I mean, obviously the fragile nucleosomes are much, they are much more dynamic in the sense that they are being reloaded or, and then removed and loaded, reloaded. So that dynamics is definitely faster because you have so many remodeling molecules acting up there. It could be one of those.
Thank you very much once again. I think we're getting close to time, so I'll pass it to Sky, I think, for the next talk. Thank you.
Perfect. Thank you so much.